Hi, my name is Jack Miller. I work with NASA Gene Lab at NASA Ames Research Center in the area of space radiation. In this talk, I'm going to give a very brief overview of some of the issues involved in studying space radiation biology on the ground and talk about some of the facilities that are available for doing so. I'll first review very briefly the space radiation environment, talk about some of the health effects of that environment on humans and some of the countermeasures that have been and are being discussed to counteract those effects. And finally, I will review some of the major worldwide facilities available for space radiation biology on the ground. There are three principal sources of radiation in space. First, the Earth's trapped radiation belts, familiarly known as the Van Allen belts. Second, solar particles, which emanate from the sun in a steady stream, but punctuated by occasional very high-intensity solar storms. And finally, galactic cosmic radiation, which originates from outside the solar system. The Earth's radiation belts consist primarily of electrons and protons trapped by the Earth's magnetic field. They extend out to about 25,000 miles, or 10% of the way to the moon. But the vast majority of human experience in space to date has been at altitudes of just a couple of hundred miles, where the radiation intensities or radiation doses are relatively low. So for missions at shuttle or ISS altitudes, so a few hundred miles up, commonly referred to as low Earth orbit or LEO, astronauts are exposed primarily to trapped particles and also some galactic cosmic rays or GCR, which penetrate the protective effects of the Earth's magnetic field. The absorbed dose is on the order of 0.2 milligray per day, and the principal hazard is so-called late effects, which would be, say, cancers, cardiovascular, central nervous system effects late in life, perhaps cataracts. The countermeasures are relatively straightforward. They're both operational, so limiting the amount of time astronauts can spend in space in total, and also the amount of time they can spend on EVAs or spacewalks, and then using the shielding of the space station itself. So on the order of a few, maybe to 10 centimeters worth of aluminum, which is sufficient to stop a great deal of the, stops all the electrons, many of the protons, although it does not stop the GCR. We'll talk about GCR a bit in a little while. On so-called exploration missions, so beyond the Earth's magnetic field, beyond the radiation belts, so missions to the moon and beyond, the hazards are somewhat different. First, there are the occasional solar particle events, which can deliver very, very high intensities of protons in a matter of hours or a couple of days, and can lead, if unshielded, can lead to acute radiation sickness, possibly disabling or possibly even fatal. And then the chronic effects, or the effects of chronic exposure to galactic cosmic rays, which again make the astronauts subject to possible late effects, cancers, cardiovascular effects, CNS effects. In this table from a paper by Simonson et al. In PLOS Biology, a recent paper from just earlier this year, you can see the typical doses for some representative missions, and I've highlighted a few of them here. If you do the arithmetic, it turns out that these all translate out to on the order of 0.2 to 0.3 milligray per day, but depending on the mission, the radiation quality is going to vary between with different proportions of protons and GCR. Now, I'm not going to talk much about solar energetic particles, or SPE, for reasons that will become clear in a moment. But just for completeness, I'll point out that they consist mostly of protons with a small admixture of alpha particles, helium nuclei, and then HCE, or highly charged and energetic nuclei. So ions with a charge greater than 2 and energies in the few hundred MeV per nucleon range. And once again, the hazard from these particles is that if you encounter a solar storm in a lightly shielded or unshielded environment, a human being would be subject to acute radiation sickness. The reason I'm not going to discuss SPE in depth for purposes of this talk is that they are relatively easy to design countermeasures for. One thing that one can do is schedule missions for the least active parts of the solar cycle, of the 11-year solar cycle. And in the event that an SPE is encountered, exploration spacecraft will likely be designed with so-called storm shelters, so very heavily shielded parts of the spacecraft to which the astronauts can retreat for 2 to 3 days when it takes for an SPE to subside. The greatest challenges facing space radiation biology investigators have to do with galactic cosmic radiation, or GCR. GCR are atomic nuclei spanning the entire periodic table. They're produced in supernova explosions, such as the one depicted here, false color image taken with the Chandra X-ray Observatory. In this log plot of the abundance of GCR nuclei as a function of atomic number, you can see that the abundance really drops off the edge of the table above iron. There are actually interesting astrophysical reasons for this, which unfortunately I don't have time to go into now. And as I already said, the hazards of these GCR nuclei are late effects, cancers, CNS, cardiovascular. On a microscopic level, the sort of effects that one is concerned with are so-called direct effects, where a heavy charged particle can produce single or double strand breaks in DNA, which might subsequently be unrepaired or, what's worse, misrepaired. And, for example, indirect effects, such as production of hydroxyl radicals in aqueous matter. So dealing with space radiation is a multi-factor problem. One has to consider the type of radiation found in space, the particular organ that's going to be affected, the various possible endpoints, including cancers, cardiovascular, and central nervous system, what countermeasures are available, such as radioprotectants, adding shielding, and planning missions, mission planning such as when you go, how long you go, how long you stay. Now, turning to the discussion of facilities, here's another plot that's a bit hard to read, but it's a plot of the abundance of several different galactic cosmic ray ions as a function of energy. And if you squint, you can see that it peaks in the area of the region of a few hundred MeV per nucleon. And as it happens, and why that's the case is an interesting subject in its own right that we don't have time to discuss here. As it happens, the green shaded region is the region that's covered by earthbound particle accelerators. The first of these accelerators that I'm going to discuss, and arguably the most important one for those of you in the U.S., is the NASA Space Radiation Laboratory at Brookhaven National Lab on Long Island. And as you can see, the red circle indicates where the NSRL is. It's part of a much larger accelerator complex. It provides ions with charges from protons all the way up to charge 90, energies up to 1500 MeV per nucleon, actually up to 2500 MeV per nucleon for protons. Doses from fairly small doses of 0.1 milligray up to 1 gray and somewhat higher. It provides a GCR simulator, which I'm going to discuss a little more in a couple more slides. An SPE simulator, so simulating solar storms. The nominal beam size is about 20 centimeters square. They can go smaller and they can also go larger, up to about 60 centimeters by 60 centimeters, with pretty good uniformity. So those beam spot sizes are sufficient to irradiate multiple animals or flasks at one time. And this is a picture, it's a rather old picture of the NSRL target area, but it's to give you an idea of the scale of the area that you have to work with, in which you have to work. And the NSRL offers a diverse array of beamline hardware for supporting biology experiments. Now, the NSRL is the only accelerator facility in the U.S. That can study space radiobiology across the whole spectrum of, or the important part of the spectrum of GCR. But there are accelerators elsewhere. I'm going to briefly discuss two of the more prominent ones where space radiation biology research is being conducted. There's the HIMAC, the Heavy Ion Medical Accelerator in Chiba, Japan, and just indicated on this plot here, or on this graphic, where their experimental area is. They offer an ion energy manifest that's not quite as diverse as the NSRL, but it still covers a significant part of the galactic cosmic ray spectrum. And they do offer beam time to international investigators, and I've indicated here the URL for getting started with if you want to look at what's available at HIMAC. The CIS-18 accelerator at GSI in Darmstadt, Germany, has capabilities comparable to those at the NSRL. They offer uranium up to 1,000 MeV per nucleon, neon up to 2,000 MeV per nucleon, and ions in between, and 4 and 5 GeV, or 4,000 to 5,000 MeV protons. And of course, they have extensive biology laboratory facilities for supporting experimenters. And many ESA scientists are conducting space radiation biology work at GSI. Now, although in this talk I focused on heavy ions, protons are still of interest, certainly, in radiation biology. And there are many proton facilities worldwide. I should note that both the heavy ion and proton facilities have been built principally for radiotherapy purposes, but they do offer, in most cases, many cases, beam time for basic radiobiology research. So what are the requirements for a facility for simulating space radiation on Earth? Well, you want to be able to reproduce the types of particles found in space, and accelerators can do that. You want to reproduce at least a substantial part of the energy spectrum. Accelerators can do that. You want to be able to at least approximate a mixed field exposure, meaning that you want to simulate what the situation is going to be in space, where organisms are going to be struck by particles of many different energies and many different charges close together in time. And the scientists at the NSRL have done some very clever things, putting together their so-called GCR simulator for approximating mixed field space-like exposures. You want to be able to generate or expose samples to doses on the order of what they're going to see on exploration missions, and that's a relatively easy one, because although individual particle hits can be very damaging, the total absorbed dose that organisms are going to see in space is relatively low, even on an extended mission to Mars. The tough one is the dose rate. You might recall from that table I showed earlier, the typical mission dose rates are on the order of, even for exploration missions, around the order of 0.2 milligray per day to 0.3 milligray per day. Accelerators are designed to operate at much higher intensities. So right now, the minimum dose rate one can get at an accelerator is on the order of 0.1 to 0.2 milligray per second. So one is confronted with a problem. So I'm going to leave you with a couple of topics for further discussion. Um, just two of many things that will be discussed in these town meetings, I'm sure. Uh, First, how important are dose rate dependent effects? Well, we know that the time scale certainly is important for, say, DNA repair and bystander effect. There may be effects where dose rate is less important. Um, but assuming that one does want to be able to more closely replicate space radiation dose rates than is possible right now, how does one go about doing that? Well, you can use, say, for example, microbeams and um, hit samples with single doses at the lowest possible intensity. You can fractionate with low doses over long periods of time. Um, these each have feasibility issues, um, and they are a subject of ongoing discussion in the community. This was a necessity, a very brief and very brief overview. Um, but since it's been recorded, I encourage you to go back and take a closer look at the slides, including the URLs for the radiation facilities that I discussed. You can get much more information from those websites, including information about how to apply for beam time. I've got a short list of suggested references here, and of course, there are hundreds, if not thousands, of references in the literature on these subjects. The ones I've selected here are relatively recent ones that discuss the NSRL GCR simulator and also give examples of research that's been done at the NSRL, at GSI, and at HIMAC. Thank you, and good luck with your research.